this chapter, we're going to discuss blood cells, which in blood cells, macrophages system, and lymphatic system. So this chapter mainly focuses on red blood cells, anemia, and polycythemia. Let's start with red blood cells. Red blood cells are also known as erythrocyte, which transport hemoglobin, which in turn carries oxygen from lungs to the tissue. So hemoglobin circulate as a free protein in plasma, and it is not always enclosed in the RBCs. RBCs have also other function beside transport of hemoglobin. For instance, they contain large quantity of carbonic anhydrase. Carbonic anhydrase is an enzyme that catalyzes the reversible carbon dioxide and carbonic acid reaction. What happened? It makes it possible for water of the blood to transport enormous quantities of carbon dioxide in form of bicarbonate from the tissue to lungs, where it is recovered to carbon dioxide. Hemoglobin is an excellent acid-base buffer and are responsible for most of the acid-base buffering power of the whole body. So, because RBCs contain hemoglobin and hemoglobin is the acid-base buffer, RBCs are responsible for acid-base buffering power of the whole blood system. Shape and size of RBCs: they are bioconcave disc having a thickness of about a diameter of 7.8 micrometer and thickness is 2.5 micrometers. shape of RBCs can change remarkably as they squeeze through the capillary. So when they are moving around in your body, they can change shape and sizes to just fit into the capillaries. Concentration of RBCs in the blood: person living in high altitude will have greater number of RBCs. We're going to discuss this later. Hemoglobin concentration in the cell: when hemoglobin formation is deficient, the percentage of hemoglobin in the cell may fall considerably below this value, and the value of RBCs may also decrease because of diminished hemoglobin to fill in the cell. So here they say that the concentration of RBCs or the blood in the human men or women is plus minus 300,000 cubic micromilliliter. Production of red blood cells: area of the body that produce red blood cells in embryonic life is yolk cell, and when you reach the gestation phase, it is the liver. Spleen and lymph nodes are also responsible for producing RBCs. After gestation or after the birth, RBCs are exclusively produced in the bone marrow. Bone marrow of essentially all bones produces RBCs until a person is about 5 years old. This RBCs continuous production in bone marrow. RBCs are continuously produced in the bone marrow, such as the vertebra, sternum, ribs, and ilia. These are the marrow membranous bones, so they also produce the RBCs. Genesis of blood cells: this diagram here shows the genesis of normal RBCs and the characteristics of RBCs in different types of anemia. So what happens is, erythroblast divides and multiples time to produce the basophil erythroblasts. These contain they can be stained with dyes. Then they form the polychromatophil erythroblast, and then they form the orthochromatic erythroblast. Polychromatoerythroblasts contain condensed nucleus, and orthochromatic erythroblasts contain endoplasmic reticulum. Here endoplasmic reticulum is reabsorbed and they form the reticulocytes, which performs diapedesis, and the erythrocytes are formed. So this is the genesis of RBCs pathway: proerythroblast, then basoerythroblast, polychromatophil erythroblast, orthochromatic erythroblast, reticulocytes, and erythrocytes. When looking at the genesis of blood cells, we must know that pluripotent hematopoietic stem cells are the cells from which all the cells are derived in the circulating blood. The intermediate cells are very much like pluripotent stem cells. Even though they have already become committed to a particular line of the cell, they are called committed stem cells. Committed stem cells then will produce erythrocyte, which is also called the colony forming unit of erythrocytes. Let's look at this diagram. This diagram states that it shows the formation of multiple different blood cells from original pluripotent hematopoietic stem cells in the bone marrow. So here you can see that pluripotent hematopoietic stem cell will produce the colony forming unit in the spleen. This colony forming unit CFUS, S means spleen, will divide further into CFUB, CFUGM and CFUM. CFUB means colony forming unit blast. Colony forming unit spleen will divide into will produce colony forming unit blast, colony forming unit granulocytes or monocytes, and colony forming unit megakaryocytes. CFU blast will produce erythrocytes. CFU granulocytes and monocytes will produce obviously granulocytes, which include your neutrophils, eosinophils and basophils, and monocytes, which in turns will produce macrocytes. And then colony forming unit megakaryocytes will produce megakaryocytes, which will form the platelets in your body. Pluripotent hematopoietic stem cells also give rise to LSCs, which are your lymphoid stem cells. This lymphoid stem cell will produce your T lymphocytes and B lymphocyte, which will help you in the immunity. Another important point is a colony forming unit that forms granulocytes and monocyte have the designation CFU, GM and so forth. So growth and reproduction of the different stem cells are controlled by multiple proteins called growth inducers. Internuclein 3 promotes growth and reproduction of virtually all different types of committed stem cell, whereas other induce growth of many specific types of cell. These growth inducers will promote growth but not differentiate of the cell, which is the function of another set of proteins called differentiation inducers. So in your stem cells, either they can be growth inducers or the differentiation inducers. Growth inducers will help you in growth and reproduction of different stem cells, but differentiation inducers will have different function. They can cause one type of committed stem cell to differentiate one or more steps towards a final adult blood cells. Formation of growth inducers and differentiation inducers is controlled by factors which are outside the bone marrow. Infectious disease cause growth, differentiation, and eventual formation of specific type of white blood cells that are needed to combat each infection. Let's see the stages of differentiation of red blood cells. So this is where we say genesis of RBCs. These are the stages of differentiation in the red blood cells. This stage, we have discussed this earlier, right? Reticulocytes. This stage, reticulocyte is called this because it contains a small amount of basophilic material consisting of remnant of Golgi apparatus, mitochondria, and few other cytoplasmic organelles. During this reticulocyte stage, the cell passes from bone marrow into the blood capillaries by diapedesis. This diapedesis is squeezing through the pores of capillary membrane. So the process of squeezing of RBCs from the bones of from the membrane, capillary membrane, from the pores of capillary membrane is called diapedesis. Erythropoietin regulates RBC cell production. Total mass of RBCs in circulating system is regulated within narrow limits, and thus one should have an adequate number of RBCs available to provide sufficient transport of oxygen. And number two, cells do not become so numerous that they impedes the blood flow. Tissue oxygenation is most essential regulator of RBCs production. Condition that decreases the quantity of oxygen transport to tissue ordinarily increases the rate of production. So if you have oxygen will decrease, the production rate will increase. At very high altitude, where the quantity of oxygen in the air is greatly decreased, insufficient oxygen is transported to the tissue and RBC's production is greatly increased so that more oxygen is available to the body. Various diseases of circulation that decreases the oxygen blood flow, particularly those that cause failure of oxygen absorption by blood as it passes through the lungs, especially lungs, can also increase the rate of RBC. So factor that decreases oxygenation are here, as you can see in the diagram, is low volume blood volume, anemia, low hemoglobin, poor blood flow, and pulmonary diseases. So these are the 5 factors that decreases your oxygenation. Okay, remember, erythropoietin stimulate the red blood cells production, and its formation increases the response of hypoxia. So if there is no erythropoietin, hypoxia has little or no effect to stimulate the RBC's production. Hypoxia is a condition when there is a deficiency of oxygen in red blood cells. Erythropoietin is formed mainly in kidney. About 90% of erythropoietin is formed in the kidney, and remainder is formed in the liver. Renal tissue hypoxia lead to increased tissue level of hypoxia inducible factor 1, which serve as the transcription factor of large number of hypoxia inducible gene, which includes erythropoietin gene. HIF1 binds to hypoxia response element residing in the erythropoietin gene, inducing transcription of messenger RNA, and ultimately which will increase the erythropoietin synthesis. What happens is, at times hypoxia in other part of the body but not in the kidney stimulates kidney erythropoietin secretion, which will suggest that there might be some non-renal senses that sends an additional signal to the kidney. Erythropoietin stimulates the production of erythroblasts from the hematopoietic stem cells. New RBCs appear in the circulating blood until 5 days later. So it takes new red blood cells to form. The time it takes new red blood cells to form is 5 days. The important effect of erythropoietin is to stimulate the production of erythroblasts from hematopoietic stem cells in the bone marrow. So in addition, once the pro-erythroblasts are formed, erythropoietin will cause these cells to pass more rapidly through different erythroblastic stages than normally they do. In the absence of erythropoietin, few RBCs will be formed by the bone marrow. Maturation of RBC cells requires vitamin D, B12 and folic acid. Vitamin B12 is known as cyanocobalamin. Vitamin B12 and folic acid are two vitamins which are very essential in final maturation of the RBCs. These are essential for synthesis of DNA because they form the because each in different way is required for the formation of thymidine triphosphate, which is the essential building block of DNA. Erythroblastic cells of the bone marrow, in addition to failing to proliferate rapidly, produce mainly larger than normal RBCs called macrocytes, and cell itself has flimsy membrane and is irregular large oval instead of usual bioconcave. Deficiency of these vitamins, vitamin B12 and folic acid will cause maturation failure in process of erythropoiesis. Maturation failure caused by poor absorption of a vitamin B12 from gastrointestinal tract, which will lead to pernicious anemia. A common cause of RBC's maturation failure is when the body fails to absorb vitamin B12 from GIT tract. This situation often occurs in the disease called pernicious anemia or atrophic gastric mucosa, which will fail to produce the normal gastric secretion, and then vitamin B12 won't be able to get absorbed in the body. Parietal cells of gastric gland secrete glycoprotein called intrinsic factor, which combine with B12 and makes B12 available for the absorption. So if there is no secretion, this absorption won't take place. What happened? How this absorption stage is available? First, intrinsic factor will bind tightly with vitamin B12. In this bound state, the B12 is protected from digestion by GIT secretions. Then, still in bound state, intrinsic factor binds to a specific receptor sites on the brush border membrane of the mucosal cell in ileum. Number 3, vitamin B is then transported into the blood during the next few hours by process of pinocytosis, carrying intrinsic factor and the vitamin together through the membrane. Lack of intrinsic factor therefore decreases the availability of vitamin B12 because of the faulty absorption of vitamins. It is maturation failure caused by folic acid or petroyl glutamic acid deficiency. Folic acid is normal constituent of green vegetables and some fruits. It is easily destroyed during cooking. People with GIT absorption abnormalities such as frequent intestinal disease called sprue will have often occurring small intestinal disease called sprue, often have serious difficulty absorbing both folic acid and vitamin B12, and thus they will lack the maturation of RBCs. Hemoglobin formation. Synthesis of hemoglobin formation begins in the proerythroblast and continues even into the reticulocyte stage. What happened? This figure here shows basic chemical steps in formation of hemoglobin. So, two succinyl coenzyme A will combine with 2 glycine molecule and will form a 4 pyrrole molecule protoporphyrin 9, which will then combine with iron forming heme, and then heme will combine with polypeptide, which will form the hemoglobin chain alpha or beta, and then 2 alpha chains and 2 beta chain will form a one single hemoglobin molecule. So, a globulin synthesized by ribosomes forming a substituent of hemoglobin is called a hemoglobin chain. There are several slight variations in the different subunit hemoglobin. The different types of chain are designated as alpha chain, beta chain, gamma chain, and delta chain. Hemoglobin A is the most common hemoglobin in the human adult. It is the combination of 2 alpha chain and 2 beta chain and weigh about 64,458. Each hemoglobin chain has heme prosthetic group. 4 hemoglobin chain in each hemoglobin molecule. One finds 4 iron atom in each hemoglobin molecule. For instance, in sickle cell anemia, the amino acid valine is the substituent of glutamic acid at one point in each of the 2 beta chains. When this type of hemoglobin is exposed to low oxygen, it will form elongated crystals inside the RBCs that are sometimes 15 micrometers in length. Hemoglobin can also combine reversibly with oxygen. Oxygen does not combine with 2 positive bonds of iron in hemoglobin molecule. Instead, it binds loosely with one of the so-called coordination bonds of iron atom. This bond is extremely loose, so combination is easily reversible. What happens? Oxygen does not become ionic oxygen but is carried at molecular oxygen to the tissue. Let's look at iron metabolism. Iron is the most important for formation not only of the hemoglobin but also the other essential elements in the body like myoglobin, cytochromes, cytochrome oxidase, catalases, peroxidases. How iron is transported and stored in the body. When the iron is absorbed from the small intestine, it immediately combines in blood plasma with beta globulin apotransferrin to form transferrin, which is transported in the plasma. The iron is loosely bound in the transferrin and consequently can be released by any tissue cells at any point of the body. The excess iron in the body is deposited, especially in liver, hepatocytes, and less in reticuloendothelial cells of the bone. In the cell cytoplasm, iron will combine mainly with protein apoferritin to form the ferritin. A ferritin is a storage ion. Smaller quantities of iron in the storage pool is an extremely insoluble form called hemosiderin. When the quantity of iron in the plasma falls low, some of the iron in ferritin storage pool is removed easily, and then it is transported in form of transferrin in plasma to the areas of the body where it is needed. When RBCs have lived their lifespan about 120 days and are destroyed, the hemoglobin will be released from the cells and is ingested by monocytes macrophage cells. There iron is liberated and it is stored mainly in ferritin pool. Absorption of iron from the intestinal tract. The liver secretes moderate amount of apotransferrin into the bile, which flows through the bile duct into the duodenum. Here the apotransferrin binds with free iron and also with certain compounds. This combination is called the transferrin. This happens by the process called pinocytosis. Iron absorption from intestine is extremely slow at a maximum rate of only a few milligrams per day. Even when tremendous quantities of iron are present in the food, only small portion of iron can be absorbed. Let's look at the lifespan of RBCs, which is about 120 days. We know that RBCs circulate an average of 120 days before being destroyed. They do have cytoplasmic enzymes that are capable of metabolizing glucose and forming small amount of adenosine triphosphate. These enzymes will maintain pliability of cell membrane, maintain the transport of iron, keep the iron of the cell, hemoglobin in ferrous form rather than the ferric form, prevent oxidation of protein in the RBCs. Many of the RBCs self-destruct in spleen where they squeeze through the red pulp of the spleen. Okay, so now that we have discussed red blood cells, let's look at the conditions that are caused by either deficiency of it or either the excess, when the body have excess amount of red blood cells. Starting with anemia. Anemia is the deficiency of hemoglobin in the blood. What happened? Blood lose anemia after rapid hemorrhage. The body replaces the fluid portion of the plasma in about 1 to 3 days, but this response results in the low concentration of RBCs. When chronic blood loss occurs, a person frequently cannot absorb enough iron from intestine to form hemoglobin as rapidly as it lost. So it will give rise to the production of microcytic hypochromic anemia, aplastic anemia due to the bone marrow dysfunction. So anemia have further types. First one is blood loss anemia. When there is a lot of blood loss, the body cannot absorb enough iron. This type of anemia will give rise to a condition called microcytic hypochromic anemia. Then the second type of anemia is aplastic anemia, which is caused by the bone marrow dysfunction, when your bone marrow is not functioning properly or there is a lack of function. What happened? For instance, exposure of high dose radiation, certain toxic chemical, lupus erythromotus, or idiopathic aplastic anemia. People with severe aplastic anemia will usually die unless they are treated with blood transfusion, which can temporarily increases the number of RBCs or bone marrow transplantation. So these are the conditions where there is a dysfunction in the bone marrow and you have a condition called aplastic anemia. Megaloblastic anemia. This is the condition we discussed earlier, which is discussed in vitamin B12, folic acid, and in intrinsic factor from stomach mucosa. Understand the loss of any of these can lead to slow reproduction of erythroblasts in the bone marrow. As a result, RBCs will grow too large with odd shape, and this condition with red blood cells being too large and odd shape, it is called megaloblast. Thus, atrophy of the stomach mucosa as occur in the pernicious anemia. Because in these states, erythroblasts cannot proliferate rapidly enough to form normal number of RBCs, red blood cells that are formed are mostly oversized, have bizarre shape, and have fragile membrane. Then we have hemolytic anemia. This have different abnormalities of RBCs in hemolytic anemia. So in hemolytic anemia, hereditary spherocytosis can occur where RBCs are very small and spherical rather than being a typical bioconcave disc, and upon passing through the splenic pulp or other tight vascular beds, they will rupture very easily. Then we have in the hemolytic anemia, there is a condition called sickle cell anemia. In this condition, the RBCs will crystals elongate the cell and give it appearance of the sickle rather than a bioconcave disc. Erythroblastosis fetalis is where Rh positive RBCs in the fetus are attacked by antibody from an Rh negative mother. These antibodies make Rh positive cell fragile, leading to the rapid rupture and causing the child to be born with serious case of anemia. So these are the types of anemia. 4 types of anemia we studied are blood loss anemia, aplastic anemia, megaloblastic anemia, and hemolytic anemia. Let's look at anemia, effects of anemia on the function of circulatory system. Severe anemia. The blood viscosity may fall to as low as 1.5 times that of water rather than normal value of about 3. Greater than normal quantities of blood flow through the tissue and return to the heart, thereby greatly increasing the cardiac output. Moreover, one of the major effects of anemia is greatly increased cardiac output as a result, as well as increased pumping workload on the heart. The increased cardiac output in the person with anemia partially offsets the reduced oxygen carrying effect of the anemia. During exercises, which greatly increase the tissue demand for oxygen, extreme tissue hypoxia results and the acute cardiac failure may ensue. Polycythemia. 2 types of polycythemia. One is secondary polycythemia and polycythemia vera, or also known as erythemia. Secondary polycythemia condition where whenever the tissue become hypoxic because of too little oxygen in the breath air, produce large quantities of extra red blood cells. This condition is called secondary polycythemia, and the RBCs count commonly rises to about 6 to 7 million millimeter, about 30% above the normal level. A common type of secondary polycythemia is called the physiological polycythemia, which occur in the native people who live at the higher altitude. Polycythemia vera. In this condition, physiological polycythemia, a pathological condition known as polycythemia vera, exists, in which RBCs count may increase to be 7 to 8 million millimeter. At the hematocrit may be 60 to 70% instead of the normal 40 to 45%. This type of condition is caused by genetic aberration in the hemocytoblastic cells that produces the blood cells. The blast cells no longer stop producing RBCs when too many cells are already present. In polycythemia vera, not only does the hematocrit increases, but total blood volume also increases. Sometimes it can be almost twice the normal size. As a result, the entire vascular system become intensely engrossed. Also, many blood capillaries become plugged by the viscous blood. The viscosity of the blood in polycythemia vera sometimes increase about normal of 3 times the viscosity of water to 10 times that of the water. What is the effect of polycythemia on the function of circulatory system? Blood flow. Because the greatly increased viscosity of blood in polycythemia, blood flow through the peripheral blood vessels is often very sluggish. In accordance with factors that regulate return of the blood to the heart, increasing blood viscosity decreases the rate of venous return to the heart. Conversely, the blood volume is greatly increased in polycythemia, which tend to increase venous return, and actually the cardiac output in polycythemia is not far from normal because these factors more or less neutralizes each other. A person with polycythemia vera ordinarily has a ruddy complexion with bluish tint on the skin. Bluish tint is also known as cyanotic skin. This brings us to the end of RBCs, anemia and polycythemia, which is chapter number 33 of Guyton and Holtz. Thank you for watching. If you like my video, don't forget to subscribe for more.